students. In last lecture, we have been discussed about the cell and molecular biology and cell organelles as well as prokaryotic and eukaryotic cells and distinguishing characteristics of both the cells prokaryotic and eukaryotic cell. Today, we will go through the study of virus as per our syllabi, for TYBSc. We will go through the study of virus. In this COVID-19 pandemic situation, hazardous situation, we all know about the viruses as well as till to the TYBSc. You have been studied or read about the viruses. But today we will go through the outline as well as some deep study about the viruses. First of all, viruses are not the living cells. So, at the time of the observations of the various microorganisms into the studies of the various scientists, first of all, Mayer have been observed in 1886. Mayer have been observed into the tobacco plant, that is the specific disease caused as the wilting of the leaves, as well as the leaves are dead, as well as they are becoming the mosaic structure onto the plant or the leaves of the plant, and he observed that it may be a bacterial disease, and he observed that there is some bacterial disease caused the tobacco mosaic virus. So that is called as the TMV. But Mayer concluded that it is the bacterium. So after that, in 1892, Ivanovsky have been observed, or further studies of the Mayer have been observed that whatever the Mayer have been got the extracted the sap from the mosaic diseased plants of the tobacco, and then he injected that juice into the infected plant juice into the healthy plants. So further studies of Ivanovsky, it was the Russian scientist, he have been observed that there is the observations of the Mayer have been tobacco plants, there is a variety of bacteria may be failed to produce, that different bacteria have been inoculated and they have been introduced into the healthy plants, but bacteria have not formed the mosaic disease into the tobacco plant. So that Ivanovsky confirmed that the observations of the Mayer that there is some specific organism which caused the mosaic disease. Okay, so at that time, after the bacterium filtration, at the time of the observations of the bacteria or the studies of the bacteria, there is a specific filtrations are there, and in the filtrations, the bacterium, they have been filtered that plant juice, and then it can be introduced to the new healthy plants, though there is the tobacco mosaic disease caused to the healthy plants by the observations of the Ivanovsky. So he confirmed the observations of the statements of the Mayer in 1886, and in 1892 Ivanovsky observed that after the filtration of the bacterium culture and the whatever the sap after the filtration that introduced to the healthy plants, it also showed the mosaic disease, okay, into the healthy tobacco plants. So that there is after that further studies, specific scientists as D. Hareley in 1917, D. Hareley have been first of all observed the bacteriophages. That is, what do you mean by the bacteriophage? That is those viruses that are infect the bacterial DNA and they are multiplied into the bacterial cell. So there is further studies have been observed by the various scientists. Then Schlesinger scientist in 1933, he have been first of all demonstrated the composition of the viruses. What is the composition? That till to the 1935 the scientists have been come to the conclusion that viruses are not the living cells, they are not the cells. Okay, so they have been concluded that viruses, what do you mean the virus? Virus, that is the poisonous or the venomous microorganism, which are not organism but the microstructure, which are causing the poisonous or the deadly disease. So they have been in 1933, Schlesinger have been first of all observed or the stated about the composition of the viruses. That composition of the viruses, you have been, that will be state that there is a composition of the viruses is protein and DNA into the virus structure. That is the demonstrated that the crystals that retain into the viral that germ, we can say, and in that there is a protein and DNA have been formed. After the Louis Pasteur and the various Robert Koch-like scientists, they have been observed the bacteriums, germs, but these are the after the further observations of the Mayer, Ivanovsky and D. Heller, etc. scientists. So it has been demonstrated that there is some specific structures which are harmful for the infectious or deadly or poisonous like disease causing to the living beings, we can say. Okay, so that in 1952 Hershey and Chase studies that bacteriophage, that is demonstrated genetic information which have been transferred to the bacterial DNA, and then bacterial DNA information transferred, that will be infection, results into the penetration of the viral DNA into the cells of the bacteria, so that there is viruses are infective microorganisms that show the several differences from the typical bacterial cell. They are different from the typical bacterial cell, so that these are the infectious. It has been concluded that, and so from these observations, it has been concluded that the viruses are not the cellular structures. So these are the very minute structures which are having the, as the Hershey and Chase scientists have been demonstrated in the 1935 that these are crystallized structures which are consisting or composition of these structures, that is the protein and DNA first of all, and then the viruses are not the cells or the living organisms. So according to the today's situation, you have been read or observed that or heard that or read that there is coronavirus, COVID-19, that is consisting with the outer covering or outer envelope. It is provided by the specific kind of the proteins, and they are protecting that crystallized structure of the virus, and so that it is not the destroyed, it not the curable. So like this, the studies of viruses, if we observe about the study of virus, that there is a, first of all, what is the size of the viruses? That is, size is very minute. That is according to the bacterial cell. It is not seen by the naked eyes. As the size is concerned, the bacterial size we can observe under the microscopes, but the viral size, that is the viral size, is it can also be observed under the microscope, but the size, as far as considering the size of the viruses or the crystallized structure of the viruses, very minute or very smaller than the bacterial cells. Okay, and so that these are not the actual cells, these are not covered by the plasma membrane and all the things of the which we have been studied earlier in last lecture. There is the cell of the any living organism, it is having the specific plasma membrane. As the bacteria, concerning bacteria, that is the prokaryotic cell, there is presence of the cytoplasmic membrane, cell wall, as well as the capsule outside the outer periphery. There is presence of the capsule. So that what do you mean by the capsule? Capsule is nothing but the outer envelope consisting with the specialized proteins which are protecting the any hazardous conditions to them, to the bacteria. As like in the viruses, there is no any wall, no any membrane is present. There is only the capsule structure, and there is a proteinous outer covering or the envelope. So that according to the size of the viruses, it cannot be multiply outside the cell. Viruses cannot multiply outside the cell. As you know about the COVID-19, if it will be transferred to the any substratum or any object, there is the living period or the whatever the activity of its viruses crystal structure, that activity is based on that substratum type. So according to the into the heat or into the sun rays, it can live up to the 3, 4 or 6 hours. But if it will be laid onto the specific substratum, that is plastic or that is iron handles of the doors, etc., then it can be live up to its limit is living up to the 12 to 24 hours. So that crystal is not living, but it is not the organism or it is not the cell, but it can be actively participate or actively multiplies into the by getting the specific kind of the environment or specific kind of the cell where it can be multiplies. It can be maintain its genetic matter, maintain its genetic structure, and it will be multiply into that after the protein synthesis. It can be multiplied into the library or the multiplication of the viruses. So that the size of the bacteria as compared to the bacteria, the size of the virus is very minute, as well as a very smaller virus size is up to the 200 angstrom units. 200 angstrom units, it is the very lowest size of the viruses. Okay, so as far as considering the viruses size, there is no any conclusion that these are very minute or very smaller than the bacterial cell, and these are not the cell. Okay, then as far as concerning the metabolism of the viruses, there is no independent metabolism of the viruses. Viruses are not metabolite themselves. Okay, they are getting the specific substratum, the specific cell to the multiply, so that they are multiply into the another cell with the help of the another cell. So no virus been cultivated into the cell-free, and cell-free medium. Into the cell-free medium, viruses cannot multiply. So that as the COVID-19, it multiplies with the one person to the another person according to it getting the cell medium. Okay, so that these are not multiply or metabolize. Their metabolism have not been shown the independent metabolism. They are metabolically inactive outside the host cell. They are metabolically inactive outside the host cell, and so that the viruses are obligatory intracellular parasites, we can say. Viruses may be considered as the obligatory intracellular parasites. Okay, so that because of this fact, viruses have been considered as the non-living. Okay, viruses are considered as the non-living. So that is the next one. After the size, there is a structure of them. There is a simple structure of the viruses. There is simple structure of the viruses, that viruses have a very simple structure. They consist of nucleic acid core. At the core, there is a nucleic acids, and that will be covered by the outer envelope, that is the proteinous envelope. And that very simple structure, that typical that cells which are made up of proteins, that respect to the differ. They are as the typical cell we can observe, there is a specific protein structure is there. And then as per concerning the structure, there is membranous envelope consisting of protein, lipid, carbohydrate outside the usual protein coat, but the envelope derived from the host cell. That is, thus essentially viruses are larger nucleoprotein particles. Viruses having a simple structure and they are larger nucleoproteins having which they are getting from the host cell and they are multiplying. So that there is no self metabolism. They are very simple in structure. At the core, only the nucleic acids are there, the nucleoproteins are there, and so that they are easily multiplied into the host cell. Rather, they are not multiplying themselves. Okay, so that they are required the living cell or the host cell for the multiplication. Okay. The next point, that is the absence of cellular structure. We know very well that is the cellular structure is absence to the viruses. As we know, that is the viruses do not have any cytoplasm, any cytoplasmic organelles, any other substances as like the organelle as well as the other substances present into the generalized cell. There is a Golgi complex, ribosomes, lysosomes, all are divided into the as like the bacterium, but they are not also having the specialized structures as like the cell, and they are utilizing the ribosomes of the host cell for the protein synthesis, for the production of the nucleoproteins, for the formation of the genetic material. Okay, either DNA or the RNA. There are two types of the viruses. There are three types, but especially DNA viruses and the RNA viruses as well as the DNA RNA viruses. So that they are getting the use of the host cell ribosomes. For example, bacterial ribosome, they are getting the bacterial ribosomes and they are multiplying or they are synthesize the proteins. Okay, so that there is a absence of cellular structure. So as far as concerned the nucleic acids, the nucleic acids are in the form of the DNA or RNA. So that they are differentiate, that is the DNA viruses and the RNA viruses. But as the Rous sarcoma virus like viruses, they are RNA, DNA and RNA, and then they are produce the protein. So that they are the DNA, RNA viruses. So that they are also called as the DNA RNA viruses. So there are different, as far as concerning the nucleic acid, there are specific kinds of the three types of the viruses are present onto the earth. So that there is specific kind of the viruses are there. As far as concerning crystallization, they may be very smaller crystallized structures are present into the virus and they are behave like the chemicals and the crystallization. So no in viruses, other characteristics of the virus is that there is no multiplication and the division. Okay, there is no multiplication division because they are not the living, we can say they are not non-living substances, and they are for the multiplication they are using the another living cell as a medium for the multiplication. So that in viruses itself themselves they are not multiplied, or there is no division into the structure of the viruses. There is the crystallized structure of the at the core there is a nucleic acids are present. Okay, so that crystallization is no growth and cell division is there, and then genetic material RNA or DNA that is capable of reproduction, that only into the host cell that is capable of the reproduction. That is, they are forming the library of the viral library into the bacterial cell. That is multiplication by using the ribosomes and all the protein synthesis process by the host cell, and so that they are multiplied. So there is no reproduction itself and there is no division of the viral structure. So these are the various characteristics of the viruses that is typical as like the living organism. As compared to the living organism, there is no typical cell division and reproduction takes place into the viruses itself. So there is characteristics that firstly that nucleic acids as a genetic material, and the nucleic acid contains that the instructions for the structure and function of the virus, and they can reproduce, they cannot reproduce themselves and using the host cell synthesis, and then there is a host cells synthesis machinery they are using. whatever the host cell synthesis machinery they are using, and they are multiplies. So according to the various structures of the viruses, there is different kinds of the structures of viruses are there. For example, polyhedral or icosahedral viruses. On the basis of the symmetry, whatever the symmetry of the structure of virus, there are three kinds of the structures of the viruses, or the three kinds of the symmetries shown or present into the viruses. That is, first, that is the polyhedral or icosahedral viruses symmetry. So polyhedral or icosahedral symmetry, that is, it may be either naked or envelope capsid. Okay, what do you mean the capsid? That is the outermost layer, proteinous layer that is covered onto the outer envelope. So there is a poliovirus or the adenovirus. These are the specific kind of the adenoviruses. That is the 5X174 virus. These are the naked viruses. Okay, they are naked viruses. And then enveloped virus, for example, herpes virus. Herpes is a disease, that is a serpent-like structure is formed onto the skin of the animals, and that is called as the herpes virus. There are different kinds of the species of the viruses also. According to the Zoster-Jepter-like viruses, herpes viruses, they are causing the skin diseases. So that there is a herpes virus is consisting with the capsid. What is the structure of the capsid of the herpes virus? There is 162 capsomeres are there. What do you mean by the capsomeres? These are the capsomeres are nothing but the protein coat or the specific capsomeres. Then we have been observed about the helical symmetry. There is tobacco mosaic virus, and the tobacco mosaic virus, that is consisting with the naked or the enveloped virus, as the influenza virus and the mumps virus. As far as concerned their hexamer and pentamer structure, there is different kind of the viruses. Then there is presence of the complex symmetry. In complex symmetry, we observe that internal structure of the virus and the external structure of the virus, that is in a complex manner or complex manner. There is constitute the head, capsid, 2000 subunits. So today we'll stop here, and in next lecture we'll study about the mycoplasma.